and then then the CO2 can go in. But actually, what triggers the potassium to go in is that hydrogen ions are pumped out into the epidermal cells that are next to the guard cells. So they actually pump protons out in large amounts. When they do that, it makes it negative inside the cell. So what causes the potassium to come in is that electrochemical gradient, the drawing of the negative charge. Okay, so when it gets negative inside the guard cell, then the potassium goes in because it's positive, and then that causes the water to follow, which makes it swell up. Does it make sense? So that's called chemiosmosis, because it's still being driven by the pumping of protons. Just stop me if you guys don't get something. Okay, co-transport, we talked about all the, yeah?
What do you mean when you say it gets negative and the potassium goes in?
Well, because the protons are being pumped out, it's more negative inside the cell than outside, so it's not really, it is negative compared to the outside of the cell. Muscle cells are the same way. Because they have more positive charges outside than in, they're slightly negative inside. And that's why when you open the gated channel in a muscle cell, the sodium flows in so quick. It's not only because it's a high concentration outside the muscle cell, but the negativity inside draws it in. So it's like an electrochemical gradient.
What is it that, like, why is it that the potassium are drawn to negativeness?
Why is it what?
And, like, you said, like, inside it becomes negative and that draws the potassium in. So why isn't the potassium drawn in instead of, like, the protons being drawn back in? Like,
yeah, that's a good question. Some of the protons, what my guess is in the guard cell, because I actually tried to look this up today, there must not be any protein channels for the hydrogen ions to flow back through. So there's like pumps to pump them out, but there's no channels for them to diffuse back in. So, but there is channels for the potassium. So the potassium flows in. And then the hydrogen ions must disperse to like the other cells around, or else it wouldn't be hypertonic inside the guard cells. Yeah?
So like how does the cell know when to pump out the H+ ions?
Okay, so that particular pump is activated by low CO2 and light. Okay, so when the CO2 gets low and the light, like in the morning, no matter what, even if the CO2 isn't going to be low, but it is, because the stomates are going to be closed all night, the light activates that pump. But it gets overridden when, even if you activate the pump, if there's not enough water in there, there's no water to flow into the cells and swell them up, so the guard cells will still be closed. So it really works by all three mechanisms. Okay? Co-transport is really important, and actually we're going to come back and talk about that as well. Co-transport, the idea is if you have like some cells side by side, in the case of, let's say this is a sugar producing cell in the leaf, and this is a phloem cell, okay? It's going to transport the sugar. What we want to do is load lots of sugar into that cell. So actually, weirdly enough, the phloem cells pump hydrogen ions into the mesophyll cell, and it binds to the sugar, and then there's a channel for both the sugar and the hydrogen ions, and then they flow back in by diffusion. That's co-transport. And apparently it's a good way to like load extra stuff into a cell. So again, that would be a form of chemiosmosis again, because the pumping of protons is fueling the movement of sucrose. But it's called co-transport a little bit more specifically because the protons are binding to the sugar, causing it to flow.
When they get back to the previous one, like wherever the protons came from, will they detach? Will it?
Yes, and then they'll pump again, and then they'll pick up more sucrose, and then it'll diffuse back through. So it's a continual process that loads up the phloem pretty effectively. Oh yeah, and then another example of that is in the roots. Actually, to get more nitrogen in the roots, the protons are pumped through the epidermis, and again, that picks up the nitrogen, and then it flows into the roots. So the epidermis of the root pumps it out into the soil surrounding the liquid part of the, the water dissolved in the soil. It picks up the nitrogen and flows back through so that the plant gets a lot of nitrogen. So why does the plant have to pick up nitrogen from the soil? This is more next week's lesson, but try to keep everything kind of tied together. Well, you know what a plant needs nitrogen for. To turn sugars into amino acids and, okay, proteins from the amino acids. What else has nitrogen in it? Nucleotides, okay? So the plant has to get nucleotides and proteins somewhere. That is why plants aren't very rich in proteins, because they don't get a lot of nitrogen. Nitrogen is the most abundant gas in the air, okay? And we're gonna go over this next time, but nitrogen in the air is that. It's N2, but it's nitrogen triple bonded to a nitrogen, and that gas is completely unusable. And there's no enzymes in plants, there's no enzymes in most animals, I guess no animals, that can convert that N2 into something like a usable form of nitrogen that can be taken up by living cells. So what happens is specialized bacteria in the soil convert the N2 into like ammonia and nitrates, which can be then taken up out of the soil. So there's not a lot of nitrogen in the soil, and the plant has to kind of super concentrate it from the soil to get enough that it can even make the few proteins that it does make. So we know it doesn't use proteins for structure, but it uses proteins for enzymes, so it has signaling and all that kind of stuff. So the plant does still need proteins, okay? So it just gets the nitrogen by co-transport. And this is the sucrose one that we already talked about. Okay, I don't think I need to remind you what osmosis is, right? Diffusion of water. Okay, our favorite. We went over this though again at midterm time, so I don't know if you want me to go through it again or not. Just the quick and dirty version. Water potential, which looks like the little Poseidon thing, is pressure potential and solute potential. Pressure potential in plants is always going to be positive, and solute potential always is negative. What is the highest it can be? 0. When is it 0? Pure water, right? Distilled water, then it's 0. Any solutes makes it negative. So the more solutes you have, the more negative water potential, the lower it is. Water always moves from high water potential to low water potential. We know water moves to where there's more solutes, okay? And because it's hypertonic, okay? Water potential becomes important because it's not, remember that pressure does play a role, and we're going to you're going to see that. We're going to talk about root pressure in a minute. We're going to talk about, again, moving stuff through the phloem. They don't have a circulatory system. Plants, they have vascular tissues, xylem and phloem, to move things through the plant, but they have no circulatory system. They have no pump, like no heart to push it around. So stuff has to move they've discovered mostly by pressure differences. So we're going to talk about that. So that's very, very important, pressure in plants. And I think, any questions about any of this junk? We've done it so many times before. Okay? And we of course remember that the cell wall exerts pressure. So even if you put a plant in pure water, the water isn't going to keep flowing in endlessly. It's going to flow in until the water potential inside and outside are going to equal 0, or they're going to be the same, and then you're not going to have any movement. Okay? So anything else you want me to go over about water potential? Because I know at midterm that was like the big question at the review is, go back over that. So we're good? Okay. This is going to be quick lateral movement, which is short distance, but we're not just talking about like diffusion, osmosis, active transport. We're still talking about movement across cells, but we're not talking about like moving from the roots up to the leaves or from the leaves down to the roots. Okay, so that's what lateral movement is. Basically, let's look at the pictures first. Let's look at this one first. Okay, basically, in a plant cell, there's considered to be 3 separate compartments. Okay. The gray is the cell wall compartment, and the yellow is the cytoplasm, and then, I know it's all square, and then this is the vacuole compartment. Somebody, of course, had to give them freaky names, so you'd have something else to memorize because they knew